Today we're going to be talking about probability, and we're going to be talking about probability in a genetic sense and how we use probability and how we use that to kind of make predictions about what's going to happen, especially when it comes in terms of crossing and mixing up genetic material. So what is probability? Probability is the likelihood that a particular event will occur. So if I were to flip a coin, every time I flip a coin, there's always a 50% chance that it will land on heads and a 50% chance that it will land on tails. Those are my odds. That's the probability. Even the term probably, right? You say, are we going to go do this after the movie? Probably. Okay, it's giving you kind of a likelihood that something will occur. And usually when we say probably, we mean maybe. But in genetic terms, probability simply means what is the likelihood that something is going to happen or something else is likely to happen, right? So the way and the reason we do this, or the reason we're able to do this with genetics, is because the way that the alleles segregate, remember those alleles are the different forms of a gene, the way they segregate is completely random, just like flipping a coin. And that's why I use that example. Is a coin flip is random, and so are the separation of these alleles. So this principle of probability can be used to predict the outcomes of genetic crosses. So just a little bit on that segregation when we're talking about that. If we have a parent that this is their genotype, okay, and we have another parent and they have the exact same genotype, what we're saying is when these parents produce offspring, they can produce offspring that look like this genetically, that look like this, or that look like this. All of them are probables, right? And then we can kind of assign a value to which one is more likely or which one are we more likely to see when we take into account the rules of probability. So that's what we mean. Like, alleles have, we have certain alleles within an individual, and then you combine two individuals and they can produce different offspring. So if you have two individuals as parents that are the same, right? They're the same and their alleles are the same, then the probability of anything else quickly drops to zero, right? Because the only offspring these parents are allowed to produce would be exactly like them, because those are the only alleles they contain, right? That's all they have. This, for example, would not be an option because neither parent contains a recessive allele. So the probabilities, and what we're going to be looking at here in just a minute, is we're going to be predicting the average outcome of a large number of events. So why would we want to do that? Why would we want to say, well, I wonder what the odds would be if we crossed 10,000 plants, right, with this trait and that trait? Well, the advantage to that is we don't have to do a cross between 10,000 plants, right? We can look at the probability and say, oh, well, you know what, around 7,500 of them are going to express this trait, and 2,500 of them will express that trait. So rather than carry out all those crosses meticulously and follow them and do all those things that we would need to do, we can just look at the probability and say, oh, it's likely to be this. Okay? And we use probability and statistics all the time. We use models and those types of things. It's how we predict weather, right? We're using models and probability to say that, with this happening and that happening, the likelihood or the probability that we're going to have rain today is this percent. They're actually giving you a percentage, right? And that's a probability. You look at the outside and say, oh, there's a 50% chance of rain. Okay? So now, just like the weather, probability cannot predict the precise outcome of an individual event. So just like they can't tell you 100%, like if it's 50%, right, it either rains or it doesn't, okay? In genetics, I cannot predict each individual outcome. So for these two parents, I can't say they're definitely going to have a baby that has two dominant alleles, or they're definitely going to have a baby that has one dominant and one recessive, okay? So we cannot predict the exact outcome. We're just kind of given an overall average outcome of, every time these two individuals have a baby, let's look at the probabilities of their offspring. So, and remember too, with probability, just because one thing has happened, it does not influence the next event. So if you go back to my coin example, right? If I flip a coin 5 times in a row and it comes up heads, a lot of people would assume that, oh, well, this 6th flip, it's definitely going to be tails. And that's simply not true. Okay? Those 5 previous events, they are all individual events, and those events do not influence the outcome of that 6th event. And a lot of people make that assumption. It's one of the reasons why casinos are so successful, because a lot of people say, oh, well, it did this the 4 previous times, so this next time it's the odds are in my favor. Okay, and that's not how probability works. The statistics, the probability is always going to remain the same. It's just kind of human nature to make that jump and be like, well, now it has to be this number because last time it was that number. And those events do not influence one another. And they don't influence one another in genetics either. These 2 individuals could have 5 kids and they could all express the homozygous dominant. Or they could have 5 kids and they could all express the recessive trait by inheriting 2 recessive alleles. So with these events, the larger the number of events that we have, and by events we mean the production of offspring, the closer we're going to get to our predicted values. So with humans, it's kind of, it's difficult to do because most people aren't going to have 100 kids. But with plants and things like that, they are. So we can really kind of start to see how these predictions, how they kind of pan out. So let's take a look. We'll stick with our same alleles. We'll stick with big B and little b. And we're going to look at how we introduce probability, or how we use probability. And we use probability and how we predict these things through a tool called a Punnett square. Okay? And so a Punnett square allows us to cross 2 parents and then take a look at our results. So let's go back to our example. Let's go ahead and assign some traits to these alleles. So we'll say our dominant allele in this scenario is going to be blue eyes. And we're going to say that our recessive allele is going to be brown eyes. And this is just for this example that we're going to do. Okay? We're going to have one parent who is heterozygous, okay? And just take a moment and think about what color eyes would this parent have, okay? And we're going to cross them with another parent who has the same genotype. Both of these parents, if we look at our key here, would have blue eyes. Remember, the dominant allele covers up the recessive allele. So both of these parents would be blue-eyed, okay? When we look at a Punnett square is going to look like a window. It is, as the name implies, a square, and it looks like a window because it's divided into 4 sections, like sometimes you look into a window of a house and it's divided up like that. When you set up a Punnett square, when you're going to run a Punnett square, I always tell students to do it the same way every time. If you're setting it up the same way every time, you're much less likely to make a mistake. So I always take the first parent that is listed, so that would be this parent right here, and I'm going to put them on top of my Punnett square, okay? And then I take the second parent that's listed and I put them on the side of my Punnett square, okay? And then there's no special math skills or anything involved here. All you have to do is simply bring the letters together. So this big B drops down, this big B moves over. This big B moves over here. This little b drops down. This big B drops down. This little b moves over. This little b drops down. And this little b moves over. So now we have a completed Punnett square. Well, what does that tell us? Okay. Well, with Punnett squares, when we're doing crosses like this, each one of these boxes represents 25%. Okay, 25%. And you'll notice we have 1, 2, 3, 4 boxes. So together they represent 100% of the offspring that these 2 individuals could produce. Okay, what it's telling us is we look at the squares, we have one box that is big B, big B. So that tells us that individual is homozygous dominant. Okay, so big B equals 25%. We have 2 squares that are big B, little b, or heterozygous. So 2 out of 4, or 50% of our expected offspring are going to be heterozygous, and then 1 out of 4, or 25%, is expected to be homozygous recessive. Now, what that means is for every time these two individuals were to have offspring, it starts over, just like flipping the coin, right? So they have a baby, that baby is homozygous dominant. That does not mean that you've eliminated that homozygous dominant choice and the next offspring must be one of the other 3 remaining choices. They could have 4 children who are all homozygous dominant, or 3 who are homozygous dominant, 1 who is heterozygous, and 1 who is homozygous recessive. It's just saying that every time they have offspring, these are the odds, right? This is the probability for that particular group. So let's look at another one. Let's change our parents. We'll leave the same, okay? We'll leave blue eyes and brown eyes. And this time we're going to say that we have one parent who is heterozygous, and then we have another parent who is homozygous recessive. So they're actually going to show the recessive trait. So we'll draw our square. We're going to take the first parent that's listed and put them up at the top. The second parent, we're going to put them on the side and then complete our square. And notice that I'm always putting the capital letter first. If you ever have a capital letter, you always want to put it first. It's just how we write genotypes. Then over here, our little b's would be grouped together. And so now things change up a little bit. Half of the population would be expected to be heterozygous, like the first parent. And then the other half of the population would be expected to be homozygous recessive and show that recessive trait. So our numbers change a little bit. Okay, before we were 25, 50, 25, and now we're 50, 50. So changing those alleles around gives us a different look at how our probabilities kind of all shake out. And that's how we use probability in genetics, right? We want to look at our ratios, and then that gives us a percentage. And a lot of times, we look at it as a ratio, right? So we'll say big B was 2 out of 4, okay? So 2 out of 4. And so that's a ratio, of 1 to 2. Okay. Or if in our other instance we had 3 out of 4, right? So that's 3 to 1. Okay. So we look at ratios, phenotypic ratios are what we call them. So, our phenotypic ratio here is that we're going to project that half are going to have one phenotype and the other half will have the other phenotype. And that is basically how we use probability, and that's how it works in genetics.